Thanks for the kind introduction and welcome to our webinar. My name is Matthias Kai and I am the head of the development unit at IDT Biologika. Today, I'd like to introduce you my company and how our versatile manufacturing platforms, specifically in mammalian cell cultivation, are used for biopharmaceutical manufacturing. From this helicopter view, you can see our state-of-the-art facility, which is able to support complex manufacturing needs from early clinical development to commercial scale production. At IDT Biologika, we bring biopharmaceutical solutions to life with passion. And this isn't just a slogan, it's the foundation of how we work. Our teams are dedicated to delivering the highest quality, adaptable solutions that meet the evolving needs of our partners and patients. So let's take a closer look on how our approach can benefit to your project. In this session, we will take a closer look at the key challenges associated with biopharmaceutical development and manufacturing based on mammalian cell cultures, and how these challenges evolve along the journey from early development to clinical evaluation and ultimately commercialization. We will then introduce IDT Biologika solutions, highlighting our adaptable manufacturing platforms and our deep expertise in mammalian cell cultivation, enabling robust, scalable, and phase-appropriate CMC strategies. An important part of our approach is integrated support across the entire value chain, from early process development and technology transfer through clinical and commercial manufacturing, helping our partners managing complexity, reduce risk, and accelerate timelines. Finally, we will also share a real-world success story that illustrates how our scientific expertise and collaborative approach have contributed to successful outcomes for mammalian-based biologics. But let's start with an introduction on my company. At IDT, we bring biopharmaceutical solutions to life with passion. And this guiding principle reflects our commitment to innovation, quality, and partnership in everything we do. We are a global biopharmaceutical contract development and manufacturing organization specializing in a wide range of modalities, from cell culture and microbial-based protein to vaccine, gene and immune therapeutics, biosimilars, and our other sterile injectables. Since 2024, we are also proud to be part of the SK Bioscience family, which strengthens our global reach, but staying as a full independent CDMO company. At IDT Biologika, excellence isn't just a goal. It's the standard we upheld across every aspect of our operations. This slide highlights the pillar of our commitment to delivering world-class biopharmaceutical solutions. With over 100 years of experience in biopharmaceutical manufacturing, we bring deep expertise and proven reliability to every project. Our end-to-end support spans from early process development through clinical trials and into full commercial production, ensuring continuity and quality throughout the product lifecycle. We operate under the highest regulatory standards, with approvals from leading authorities, including the EMA, FDA, and ANVISA. This global compliance framework allows us to serve partners across multiple markets with confidence. In the past decade alone, we have invested more than 400 million into expanding and modernizing our facilities, demonstrating our long-term commitment to innovation and scalability. Our efforts have not gone unnoticed. We've received repeated honors, including CDMO Leadership Awards in 2025, recognizing our excellence in quality, reliability, and customer satisfaction. And beyond business, we take pride in our social responsibilities, creating long-term values not only for our partners, but also for the communities we serve. A closer look on our end-to-end capabilities is shown on this slide, supporting your product from early development through to commercial supply. We provide comprehensive capabilities across the entire value chain, starting with process and analytical development, followed by drug substance manufacturing for both cell culture and microbial-based systems. From there, we move into aseptic fill and finish, labeling and packaging, and finally, quality controls and analytics, ensuring that every step meets the highest standard of safety and compliance. Our expertise spans a wide range of modalities. In process development and drug substance, we work with cell culture and microbial-based proteins, including recombinant proteins, monoclonal antibodies, enzymes, and biosimilars. We also support vaccine development, covering viral, bacterial, live attenuated, inactivated, recombinant subunit proteins, and virus-like particles. In the field of gene and immune therapeutics, we handle complex vectors such as AAV, lentivirus, adenovirus, herpes simplex virus, and other viruses, providing tailored solutions for advanced therapies. When it comes to drug product manufacturing, we can round the value chain for our produced products in DS, but also offering independently filling of sterile injectables for both large and small molecules. This slide provides an overview of IDT Biologika's technology portfolio for mammalian cell-based production, reflecting both flexibility and scalability across development stages. We work with a broad range of well-established mammalian cell lines, including priority systems, as well as industry standards such as HEK 293, Vero, CHO, and MRC5, enabling us to support diverse products and regulatory pathways. The upstream processing, our portfolio spans multiple cell culture formats. Static systems such as cell stacks and hyperstacks are used primarily in early development and for small-scale clinical and commercial supply. As a scalable fixed-bed platform, the iCELLis and universal technology plays a central role, allowing straightforward scale-up for adherent systems for development and from development to large-scale manufacturing. In addition, we also offer single-use stirred tank bioreactor systems across a wide range of volumes, supporting flexible and robust scale-up strategies towards clinical and commercial production. And while roller bottles are no longer considered state-of-the-art, they remain in use for certain established products and specific process requirements where they continue to offer proven performance. Overall, this technology portfolio allows us to select the most appropriate mammalian cell culture system for each product and stage, balancing innovation, robustness, and regulatory reliability. These capabilities and experience is proven by our 100 years tradition across key therapeutic areas and manufacturing capabilities, demonstrating why we are a trusted partner for both clinical and commercial scale projects. For example, for vaccines, we have supported a wide range of reference projects, including Zika, chikungunya, dengue, RSV, HIV, Ebola, and COVID-19. We have also been a reliable partner for governmental organizations, delivering emergency use vaccines for more than 2 decades. In the area of gene and immune therapeutics, we have worked on pioneering projects involving oncolytic viruses such as herpes simplex and measles, and as well viral vector-based gene therapies like AAV. We are proud to be the exclusive CDMO partner for one of the first commercial oncolytic virus products, underscoring our leadership in advanced therapies. Our capabilities in aseptic filling of sterile injectables are equally robust. For over 35 years, we've been a trusted supplier to customers worldwide, handling both large and small molecules. Our expertise spans vials, both liquid and lyophilized, as well as pre-filled syringes. Reference projects include epoetin biosimilar, commercial medical Botox, Ebola and RSV vaccines, and gene therapeutics in clinical development. And to give you a sense of scale, we have manufactured over 4,500 GMP batches in just the past 4 years, demonstrating our reliability, consistency, and capacity to deliver. To give you a deeper insight into our manufacturing capacities or capabilities, I would like to briefly introduce the DS Building 205, one of our GMP facilities dedicated for mammalian cell culture. This site is designed for small to mid-scale production, supporting both clinical and commercial applications. It comprises approximately 900 square meters of clean room space, including Class C, B, and A and B areas, fully compliant with GMP requirements for sensitive biologics and advanced therapies. The building is organized into 4 independent manufacturing suites, which allows us to run multiple projects in parallel while ensuring strict segregation and robust changeover controls. The facility supports a broad range of modalities, including viral vector and protein production, vaccines, oncolytic viruses, and therapeutic biologics. In terms of technology, we offer flexible platforms that include cell stack or cell factory, the ICELLUS 500 fixed bed bioreactor, roller bottles, and 200-liter single-use stir tank bioreactors. This variety enables us to tailor the product approach to the specific niche of each product and scale. As with our other facilities, we also provide versatile downstream processing equipment, which can be configured to meet the unique requirement of each process. This facility is one of our key parts of our adaptable manufacturing lines, supporting the growing demand for mammalian-based biologics and gene therapies. Our newest DS building is the Building 203, built in 2023, covering large-scale mammalian cell line production capabilities and capacities designed to meet the high demand of commercial manufacturing. This facility features a 1,700 square meter clean room area classified as Class C, B, and A, and is equipped with 5 large production units, here shown in green on the floor plan, and 3 C-train rooms displayed here in blue. This setup allows us to support high volume production while maintaining flexibility across multiple projects. The DS Building 203 is purpose-built for viral vector and protein production, as well as vaccines, oncolytic therapies, and other therapeutic modalities. It's a cornerstone of our capacity to deliver complex biologics at commercial scale or higher demand for late-stage clinical development. Similar to Building 205, we have different technologies installed: cell stacks, ICELLUS 500 bioreactor, and single-use stir tank bioreactors, here at a higher range from 50 to 2,000 liters. And this operational range gives us the ability to scale processes efficiently from late-stage clinical development directly to commercial manufacturing without any further change of the production line, building, or equipment, and ensures at least a seamless transfer into market supply. This slide illustrates how IDT Biologika's platform solutions address the full range of scale and technology requirements in biopharmaceutical manufacturing, from early development to commercial supply. The different cell culture and bioreactor platforms are shown based on their typical dose range per batch. While actual doses depends on yield and dose formulation, this provides a useful high-level comparison of manufacturing potential. The portfolio is divided into suspension-based systems such as stirred tanks, wave bioreactors, and shaker flasks, and adherent-based systems including microcarrier processes, fixed-bed bioreactors like ICELLUS, roller bottles, and cell stacks or cell factories. The x-axis highlights scalability, ranging from small-scale production to large commercial batches. Some technologies have inherent scale limits, whereas stir tank systems offer essentially unlimited scalability. However, product characteristics and process requirements often drive the final technology choice. Selecting the right platform early in development is therefore critical. This is why we focus on technology consistency across development, clinical, and commercial manufacturing, enabling efficient and reliable scale-up. In short, our platforms are designed to adapt with your product as it progresses. Now, let's take a closer look in a real-world example from one of our customer projects. In this case, the customer was preparing the manufacturing concept for the transition from early clinical production to phase 3 and commercial manufacturing, which is the critical step that requires substantial process optimization. Initially, the customer had completed phase 1 and 2 clinical trial material production at a moderate scale. The former process workflow is illustrated on the right-hand side of the slide. However, as they looked ahead to phase 3 and commercial readiness, it became clear that the existing process would not scale efficiently. Several key challenges emerged during process development. The upstream process relied on CS40 technology, which limits scalability, particularly due to the centrifugation capacity constraints afterwards in the clarification step. The systems in use were not state-of-the-art, which impacted performance and efficiency. And there were also too many manual steps, reducing process control and robustness. And as a result, process validation and PPQ became increasingly difficult. And finally, achieving GMP compliance at commercial scale posed a significant challenge due to the unrobust process design. And to overcome these hurdles, we worked closely with the customer to redesign the process and transferred it to a scalable GMP-compliant technology, laying the foundation for CTM-free manufacturing and future commercial production. I selected this case to highlight the importance of early planning and the right technology strategy when preparing for late-stage development and market readiness. In the upstream processing, we supported the customer in scaling up their virus production process by introducing the iCELLis 500 bioreactor system and optimizing the seed train strategy. To address the higher cell demand for the iCELLis 500, we redesigned the seed train by integrating hyperstack 12 and 36 layer system, which allowed for a more efficient and compact expansion of adherent systems or adherent cells. The real transformation came with the implementation of the iCELLis 500 fixed bed bioreactor. This system enabled us to tightly control key process parameters such as pH, deserved oxygen, and temperature, ensuring consistent and reproducible conditions throughout the production run. As a result, we achieved a 12-fold increase in production scale while also improving process control and reducing manual handling. The final harvest volume reached 140 liters, which was formerly only 12 liters. In the downstream processing, we addressed also scalability limitations by optimizing the process design and introducing more robust technology. The original DSP relied heavily on centrifugation, which posed challenges for scalability, consistency, and GMP compliance. To overcome this, we redesigned the process to incorporate filtration and chromatography-based solutions. As shown in the flowchart, the process begins with the 140-liter clarified feed, followed by DNA removal, and then tangential flow filtration for ultrafiltration and diafiltration. Then we introduced an anion exchange chromatography step to enhance product purity and remove process-related impurities. This is followed by a second TFF step, sterile filtration, formulation, and finally filling, labeling, and storage. The process yield was 1.5 liter of final drug product after sampling, ready for clinical or commercial use. We also evaluated hold times at critical stages to ensure process robustness and mitigate risk during manufacturing. This optimized DSP design not only improved scalability and the integration of single-use process solution, but also enhanced process control, product quality, and regulatory readiness, key factors for successful PPQ and commercial launch. The result was a successful scale-up and validation of the optimized process, highlighting both consistency and comparability across production scales and technology. Let's start with the first graph on the right, which shows the titer profile across 3 large-scale IDTbiologika 500 batches. As you can see, the virus harvest titer remained highly consistent across all runs, confirming the robustness of the upstream process, which was critical for demonstrating process robustness and PPQ readiness. The second graph on the right compares virus titer at downstream stages, from harvest to formulation. The result shows that the large-scale IDTbiologika 500 process delivers comparable yields to the previous CS40-based production process. In addition to performance, the new process design introduced a simplified, closed and scalable upstream and downstream platform, significantly reduced contamination risk and aligned with modern GMP expectation. This case is a strong example on how thoughtful process redesign combined with the right technology and expertise can enable a smooth transition from clinical to commercial manufacturing. To wrap up, let me briefly explain why partners choose to work with us. To conclude, we combine innovation and technology through ready-to-use platforms and proven proprietary solutions that support efficiency and a low-risk development. This is supported by fast and seamless tech transfer, where dedicated experts stay closely involved to ensure smooth transition and reliable execution. With our regulatory excellence built on long-standing experience with global health authorities, we help to enable secure and confident market access. We also offer flexible capacity and integrated services from scalable manufacturing to in-house analytics and a secured supply chain all under one roof. At the center of everything is trusted partnership based on collaboration, transparency, and clear communication. In mammalian cell cultivation, this translates into strong upstream expertise and robust scalable process support for both clinical and commercial production. And that's how we help to bring products to the market efficiently and reliability. And with that, I'm at the end of my presentation. Thank you so much for your time today, and I'm happy to give you an oversight about our technologies. And please reach out to us if there is anything that we can do to support you.